Okay, this is our lecture on chemical reactions and enzymes. And you might be wondering why we have a picture of what clearly is a very rusty chain. Well, what happens to iron, steel, when you leave it outside exposed to water and oxygen? You get rust. And rust is an example of a chemical reaction where you take a product, which is the chain here, and you create something called iron oxide, which is the rust. If you've ever driven through Utah, you may have seen some red hills outside of maybe St. George, Utah. I was just there, and there are layers of red rock just like that, same color, and that's because there's iron in the soil. And so you literally are having layers of iron that has been oxidized and turned into a rust, or an iron oxide in the soil, and that's what makes it red. So that's an example of a chemical reaction. Enzymes are special types of what we call catalysts, and here I'll write the word for you, catalyst, that help chemical reactions run faster. Okay. And if they're working faster, they're also going to be working more easily without extra energy from the body. So that's the benefit of having these catalysts, helping chemical reactions in the body run faster, which means the body can run more efficiently. So as we move on here, chemical reactions are processes where a set of chemicals are turned into another set of chemicals. And so what we're talking about is products, and we'll just use samples A plus B, okay, or I'm sorry, reactants. So reactants A and reactants B react together in a chemical reaction to produce something called C. And anything here on the right side of the arrow is going to be a product. So in that rust sample that I was just talking about, our reactants would be the iron and the oxygen in the air and the water, and that would turn into iron oxide, your rust product. So some chemical reactions happen really slowly, and some can happen really, really quickly, like an explosion. Well, you know, if you've ever let something rust, it can take a long time. Sometimes as early as you'll start seeing rust form in a couple of days, and sometimes it, you know, for a car completely to rust away, it can take years and years. And other things happen so quickly that they release a lot of energy and you get an explosion. You don't want these kinds of reactions happening in your body. You don't want things to move as slowly as rust, and you don't want things to be exploding in your body. I think you can understand clearly how that would be life-threatening. So we need chemicals to help reactions happen faster. So here are your definitions for reactant and your definitions for products. Okay, make sure that anytime you see a word in bold, you know that those are vocabulary terms, so make sure. So again, everything on the left side of the arrow, and sometimes we have multiple reactants and multiple products, but everything over here are your reactants, and everything to the right of the arrow are your products. The arrow itself means yields, which is another fancy way of saying produces. So reactants yield products. So these two things will chemically combine to form the products. Something really important as we move forward into chemistry is if you have 10 atoms on this side, you must have 10 atoms on the other side. So matter cannot be created or destroyed, it can just be transformed. So everything that we start out with, we have to have the same amount of stuff on the other side of the equation. So what's going to happen to chemical bonds during chemical reactions? What's going to happen to chemical bonds during chemical reactions? So what we're talking about is the sharing or the trading of electrons. And I want you to keep this in mind. This is a really big, big concept. You might think about when you did your organic molecule modeling activity and you took and we put together 2 amino acids. And that looked something like this. So I'm going to draw an amino acid here, hydrogen here, hydrogen up here. So I'm going to make just a generic amino acid, so I'm showing in our group. And we get this amino acid next to the other one. You made a serine and an alanine. I'm not telling you what amino acids these are, just making them generic. So right now, these amino acids are not bonded to each other. In order for them to bond together, you must break the chemical bond here and here, and allow a chemical bond to form, a new chemical bond to form between these two atoms, and a chemical bond to form here. And so your products are water and this new double-chained dipeptide, okay, which is the formation, the starting of a protein. So let me ask you the question again. What happens to chemical bonds during chemical reactions? What did you just see me do? You saw me break some chemical bonds and then reform them in new arrangements. So let's go on in the lecture. So chemical reactions always involve changes in the chemical bonds that join atoms and compounds. And we're going to be breaking and reforming chemical bonds all the time. So we're going to be breaking bonds and forming new bonds. And those are our chemical reactions. Breaking bonds and forming new bonds. So remember, if I go too fast in this, you can, of course, pause it, go back and write down everything that I'm writing down, or listen to me and my explanations or my ramblings again. So energy in chemical reactions, energy can be released or absorbed. So if we're talking about an explosion, that's obviously a lot of energy being released. And if we're talking about rusting, that's an energy absorption. So we call these endothermic and exothermic reactions. Okay. How does energy changes affect whether a chemical reaction will occur? Okay, so let me ask you a question. If you have a list of chores to do, what are you most likely to do first? The chores that are the easiest, or the chores that take the most energy from you and are the hardest? I don't know about you, but I would like to see my list get shorter, so I might go ahead and do those easiest chores right away. And then I say, oh, look, I've already done 5 of them, and I leave the worst one for last. The things that take the most amount of energy are going to take the longest. So in chemical reaction, the more activation energy that is needed will take the longest. And so chemical reactions that release energy will happen spontaneously. That means all by themselves. They're going to happen spontaneously, just instantly. But chemical reactions that require energy need a kickstart. They need a source of energy. And we're going to look at this and we're going to learn that this is called activation energy. So when I ask my kids to clean their rooms, for example, I have to give them some activation energy. I have to help them get started. So it takes energy from me to get them kick-started. And then once I get them kick-started, then they start to work and it goes on okay. So that's like activation energy. And so here we have hydrogen gas is reacting with oxygen to produce water. And this is an energy releasing action where hydrogen gas combines with water to form two water molecules. And again, remembering that we're going to count our atoms. 2 times 2, we've got 4 hydrogens on this side and 2 oxygens on this side. And look, on this side, we've got 2 times 2, 4 hydrogens and 2 oxygens. 2 oxygens on that side. So our equation is balanced. We cannot have all of a sudden hydrogens or oxygens just disappearing. Where would they go? We don't just, matter just doesn't disappear. It has to be found. So if you ever encounter an equation that I say is not balanced, it's because we all of a sudden have an atom that mysteriously appears or disappears. In order to break water molecules apart, it takes a lot of energy. And so this is one thing that they could run electricity through this chemical reaction and make it there's electricity, and split apart the water molecule to create hydrogen and oxygen gas. But this requires energy. And this is called electrolysis. And I think that was on one of our other activities before when we talked about electrolysis. Also hair removal, right? But that's not what we're talking about. So in order to stay alive, we have to have chemical reactions that require energy, and this is why we eat food. Remember, matter and energy are conserved, and we eat plants or other animals that have eaten plants, and that's where we get our source of energy, the work that the plants have done or the other animals have already done. So, activation energy. This release of energy doesn't always happen right away. The kickstart of energy that's needed to get the reaction started is called activation energy. And here's a graph, and you drew this in your interactive notebook. So notice an energy absorbing reaction, the product is found at a higher energy level than the reactant. So this means that energy was absorbed. These types of things would get colder. If you take a Ziploc bag and you put vinegar, just white vinegar, and baking soda inside the Ziploc bag, and you allow them to mix, it's going to actually get cold inside the bag. And that's an example of an endothermic reaction. Endothermic. Here, your reactants are at one energy state, your products are at another. Energy is going to be released, so this is going to get hot. It can give off heat or light, and this is called exothermic. This is like a firecracker exploding, or when you burn gas in an engine, your engine gets hot. It's an exothermic chemical reaction. Enzymes are a special type of chemical that allow life to be possible, because without an enzyme, these chemical reactions would happen so slowly that it's really not going to be practical for living systems. Okay, so they got to be practical for living systems. So it makes these, because the activation energies are too high. It takes too much energy to get the chemical reaction started. So we want to have some type of aid, assistance in getting the chemical reaction started so that it'll be more practical for living things. And there we're introducing that term catalyst. So I want to make sure that you know that catalyst is a term that's in your book. That catalysts help make, to lower activation energies. Okay, speeding up a chemical reaction by lowering the activation energy. So why are enzymes important to living things? Well, think about it. Why is it important to you as a living thing that you clean up your room when your mom tells you to? Because you'll die if you don't, right? So if you had someone that would really help you get in there and clean up, or how about a good system of being organized, it's easier. So if you have a great system of being organized in the first place, it's not going to take a lot of energy for you to clean your room, so the activation energy is slower, and those organizational bins that you keep all your stuff in, that's the catalyst, a system. Here we're talking about molecules that act as these biological catalysts. So enzymes are proteins, okay? Proteins that act as biological catalysts. And freezing up here, okay. They speed up chemical reactions that take place in cells by lowering activation energies. So this is really important. They are proteins that lower activation energy. They speed up chemical reactions and make them happen at a speed at which allows life to really be possible. Not rusting over days, but happening within seconds, milliseconds in fact. And this has a dramatic effect on how quickly a reaction can take place. It's going to happen faster in a shorter period of time, and over and over and over again. They are very specific. They only do one thing, and they're specific because they have specific shapes. So the shape is specific, not Pacific, specific. And so its name is usually derived, we usually get the name from what it breaks down. So if you're breaking down glucose, okay, if you're breaking down glucose sugar, or let's not use glucose, let's say starch, the prefix for starch is amyl, A-M-Y-L, okay? So amyl, so the enzyme is called amylase. And a lot of times ASE will be the ending word. If you're breaking down lipids, which are fats, oils, or waxes, your enzyme is called lipase. Okay? If you're breaking down proteins in general terms, we have a class of enzymes called proteases. Okay? Okay. Enzyme actions. This is where I like to use another analogy of being like a matchmaker. If I was at a dance with you and there was, out on the dance floor here, we have a lot of people. I'll draw you a little picture, right? So this is a dance in the gym, and we have here's the gym, and you're person A over here, right? And here's this person over here that you like. And I'm in the middle. I move around because I'm supervising. Okay, that's me. And I'm going to act like the catalyst. And I walk over and I see you and I see this person and you say, I'd like to dance with that person, but I'm too shy. Well, what I'm going to do is I'm going to act like a catalyst. I'm going to get you and I'm going to get the person and I'm going to get you guys together. See, I'm going to move in here, and there's me. And I'm going to get you guys dancing. But am I going to stay with you? Am I going to dance? No, I'm going to move away, and you guys are dancing. So I'm helping this chemical reaction happen, this dancing relationship happen. And I can do this over and over and over again for other couples, okay? Like couple D and E, who are too shy. It would take them too long. Maybe it'd take you the whole night. Well, and within the first song, I can help get you guys together because I'm acting like a catalyst. Well, catalysts help chemical reactions inside of the body because they provide a place, a site, or a place where the reactants can be brought together to react. And it makes it easier for them to react. It takes less energy for them to come together. And the thing that gets worked on is called a substrate. And you should have labeled your Heartbreak Ace lab. I'm going to draw it in pink now because our Heartbreak Ace little pictures were pink. So you had your substrate was the heart. And then it's so hard to draw this. But you had the enzyme. That's horrible. Oh my goodness. Okay, and then we had the heart moving into the enzyme, and then sorry, this cold is driving me crazy. I have the sniffles. And there's your broken heart. So let's label this up. This is your enzyme here, and I'll label this E for enzyme. Notice your enzyme doesn't get changed. This is your substrate, your intact heart. Your substrate bonds here, and then these are your products. So the purpose of the Heartbreak Ace, remember ASE, the Heartbreak Ace enzyme is to break apart hearts, to break hearts. So that's why we call it Heartbreak Ace. Doesn't really exist, but it's a good analogy. So your substrate bonds with the enzyme. Now we call this position here the active site. The active site, the place where the enzyme and the substrate come in contact with each other. And then this is P for products. Now, why is it important that the enzyme stays unchanged? Well, the enzyme has to be able to do this chemical reaction over and over and over again. And so that's why. Rewind back if you didn't get all that picture. Okay? So we also call this the lock and the key. The lock and the key. Why? Because The lock has a specific shape, and only one specific key will fit into it. The key that opens the door to your house does not open the door to your neighbor's house. The substrate and the enzyme stay together, and then once the reaction is done, the substrate moves away into its products, and the enzyme can do the reaction over and over again. So here's another example using glucose and energy to produce glucose 6-phosphate, a change here. Really understanding what it is that comes in and out is not important, understanding what it is. What's important is understanding that what goes in has been changed. So notice what goes in is a glucose molecule, and what comes out is a glucose 6-phosphate. So it's been changed. The substrate has been changed. ATP goes in with these 3 phosphate groups, and what comes out is something called ADP, which only has 2 phosphate groups. So see that little blue ball there that was here, okay, is now here. So this enzyme helps to facilitate, helps this chemical reaction, this transfer of the phosphate group over. Now, hopefully you looked at your, very closely at your lesson plans that I gave you, because there was an extra credit opportunity. So you need to go back in Google Classroom and look at our lesson plans, because you're given an extra credit opportunity to do that active art, and that would be due on the day of the test if you want to get a couple of extra points, extra credit points on your interactive notebook. Or I might do it as a lab, I'm not sure. But you have to show me that you did it. And if you did it, that means you either read your lesson plans really closely, or you actually watched 21 minutes and 53 seconds of this video to know that there was extra credit available. And that means you have to go and you have to do the active art activity, and then either print out the list of questions and answer them, or you can do it all on Google Classroom, I guess, if you want to, or handwrite it all out. That's extra credit due Thursday. Okay, regulation of enzyme activity. So how are we going to know if enzymes are going to be able to work? They are influenced or affected by lots of different conditions inside of the cell. Enzymes have to work at specific pHs. So we're talking about acids and bases. If an enzyme is adapted to work in an acidic environment, you cannot take it into an environment with a high pH. It's not going to keep working. So there are enzymes in our stomach, for example, that are adapted to working. They work best at a pH of about 1. But those enzymes are not going to work in your intestines, because in your intestines the pH goes back up above 7. So they're not going to work. They're going to stop working. The same thing is with your mouth. Inside of your mouth you have about a pH of 7. It's neutral. If we take the enzyme amylase, which is in your mouth, and once you swallow and it goes into your stomach, it stops working. So that protein is inactive. Enzymes are influenced by temperature. So this is what we see happening when you cook meat. Enzymes change their shape, and we call that denaturing. Denature. Think about, remember, enzymes are proteins. An egg is a package of protein. When you apply heat to an egg, what happens to it? It cooks. It changes. It goes from that runny snot clear material and a liquidy yellow yolk to a solid white and a solid crumbly yolk. Totally changes, and that's because of a change in temperature. If you've ever eaten ceviche, which is taking seafood and spices and lots of lemon juice and onions and tomatoes, when you add that lemon juice, what's the pH value of lemon juice? It's like a 1 or 2. It's very, very acidic. What does that do to the seafood? Well, it goes in raw, and you don't apply any heat to ceviche. It just denatures because of the acidic pH, and it basically cooks it, but chemically without heat. So think about that next time you eat ceviche. So cells regulate the activities of enzymes. They can turn them on and turn them off. So if it's an enzyme like lactase, okay, lactase enzyme, We know it's an enzyme because it's lactase. Lactase breaks down lactose, which is the sugar that's found in milk. Okay, it's a disaccharide, it's a double sugar. So if lactose is present, lactase is activated and it breaks it down, the lactose. If there's no lactose around, then lactase is inactive. It isn't doing anything because it's turned off. It cannot break down anything else but lactose. Now, you guys have heard of people being lactose intolerant. These are people that get stomach aches and gas and diarrhea and bloating if they eat anything that has lactose in it, like ice cream, cheese, milk. That's because they do not have the DNA that gives them the code to make this enzyme. Without the DNA code, they cannot make this chemical protein, this enzyme. And so if they eat lactose, it makes them sick and very unhappy. So enzymes do some really important things. They regulate and control the rate of chemical reactions. They help to make the material that cells need. They release energy, and they help with transferring information. So that's the end of this lecture on enzymes. You need to do the quiz at the end of this. So go back and open up the PowerPoint in our lesson plans that I sent you, and go through and take the quiz and see if you can come up with the right questions. And then bring your questions to me tomorrow, and we'll finish our review. See you later.